is always aviremic people, right? So we're not doing sick people in this case. And what we always find is that the blood of all healthy people always has a lot of viruses in them. So we think that basically you're viremic all the time. And most of them look like these big, maybe a herpes virus or something. If you go and sequence there, what you find is that essentially everybody has TTV. So sometimes you can detect it, sometimes you can't. The herpes viruses, these guys are so common. Chlamydiophages, actually this is my favorite right here. So there's a cowpox virus in there that is really common in people, and it looks like a very derived version. This is the crystal structure of one of its things, one of its genes. It looks like it's a very old version of a pox virus that are common in people. Okay? And again, you can range from unknown viruses being 10% to 90% of the sequences you find in these things. And in this case, right, with the human stuff, we know really well that those are not human DNA because we have that to compare with. So it's really amazing out there. This is just TTV, and really these adenoviruses are everywhere. These are in everything. And Breitbart Lab has been doing a lot of that, but basically everything from sea turtles to mosquitoes to humans, they're all over the place. I should put this in here. Since you guys like plant viruses, right? The mosquitoes have a ton of plant viruses in them. They're carrying around these plant viruses. And I didn't know this, but most mosquitoes are actually sapsuckers. So if you look at them, they actually have this. What we're doing right now is we're finishing off what the normal virome is of humans and of animals. In particular, we're looking at bushmeat. And then we're trying to get to probably what I would assume you guys want to do too, really quick ways of using these techniques. And that's been working in cystic fibrosis. So we can take a cystic fibrosis sample and we can turn it around in about 2 days, and we can turn around and tell the clinician, the clinical lab, to go and culture something that they hadn't thought about. And that works nicely. So that gives you a different feel of it. The other thing that we're doing now is trying to figure out what all these stupid unknowns are, because it gets a little old, just saying we have all these unknowns. And I'm just going to show you just briefly to give you a feel of, this is a joint grant with Rob Edwards, myself, Panka Segal, and Alex Bergen. And here's the problem, all right? We've been sequencing now, so this is the first metagenome right here, all right? We've been sequencing for 1 decade. And we have the same 75% unknown problem, right? So everywhere we look, basically everything is unknown. They're not coming together. The curve is like this. And I'm actually getting tired of it. Yes.
Did you try to test again your metagenomes?
Again?
To see if the number of unknown decreased?
It does a little bit depending on the environment. Right? Some places like the marine environment is starting to compress. Other places like soil, it's just getting worse. So it does. In the marine environment, there is some compression now. We can start to see that we may be coming up to that, to the top of that curve.
So, but metagenomes that you sequenced like 10 years ago, did you try to black them again?
Yes. Yeah, exactly. That's exactly what we're doing. In fact, we're going back and we're resequencing all the samples too. It's kind of where we are. So, because now we can do so much more sequencing. And 454 will have a new 1,000 base pair system, which will be nice because then they'll be more comparable. All right, so that's bad, but it's actually worse. So back in a long time ago, we estimated that there was something like 2 billion unknown ors in that part of the thing. It's actually bigger than that. So this part of this chart is just getting bigger and bigger. Okay? Most of the variation between different environments and between different people is actually in this viral fraction, right? So we really do need to start knowing what these things are. And then there is what I call the French problem, all right? So you guys are making it worse by going out and finding all of these new things. Yeah. So these big guys, annoying. These guys here, this is Craig Billy's stuff where, the virus that grows outside the cell, that's really annoying. So he's finding all this stuff. And then of course, Costel and stromatolites found that 99% of the viruses we're finding there are unknowns. All this is problematic. So how do you actually approach it? And if you start looking at the metagenomes in general, what you figure out is that, and just for basic virology, is that really there's 3 classes of viral genes out there, one of which is really easy to figure out, and that's the viral genome replication ones, right? So those guys actually pop out really easily because they have these conserved, things for DNA polymerases and RNA polymerases and primases and so forth. And the packing genes turn out to also be easy. If there's something conserved in packing genes, you can see that. It doesn't really matter if they're eukaryotic or phage. Structural proteins, on the other hand, are really hard. So you can take where you know that the structural gene is where it's located because you can see where it should be, and you still can't identify it bioinformatically. And the way that you get around that is with proteomes. And then finally this is the one that we're all interested in here, and these things are going to be very hard. So this is one way that we're going about doing this right now, and that's basically by coming up with better annotations and predictions. And that means you don't always use BLAST. So we'll collect the viruses, concentrate them, clean them up again, and now we're doing metaproteomes at the same time we do the metagenomes. Okay? And then we take the peptides here and we map them onto the ORFs that we get here, and we're able to identify the viral structural proteins from a community. Okay? And then we train neural nets with these genes and ask, what do we have? And this has worked out really nicely because something that I didn't know that you can actually do. So if you just take an ORF that you predict is a structural protein, in lots of cases you can just synthesize it, just have it made for you. Then you clone it, express it in coli, and then look for the structure in EM. And it's been working really beautifully. So you can see you get the capsid proteins and you get the tail fibers, and it works nicely. And you can do this from things that are completely unknown, so things that bioinformatically have no hits, and we're starting to figure this, get this to work. So the idea will be that we'll compress this part of the space that we have to search through. The other one are the virally encoded metabolic genes. And what we've been doing here is you take the unknown ORFs, you eliminate the structural ones, right? And in this case, we're doing ocean stuff. Then we just synthesize the genes, express them in coli, and grow them in chemostats, and then we're doing metabolomics and custom biolog arrays on them. And this is just an example. This is one of the ones that we've been wandering around forever. It's called FOH. It was originally described as a gene that was upregulated in E. coli when you starved it of phosphate. Okay. And it's very common in the world's oceans, not really in the human stuff, but very common in the world's oceans. So we wanted to know what this is, and we actually saw it way back in like 2000. Never knew much about it. So it seems to have worked. So what we do is we're doing these unknown ORFs, synthesizing, expressing, and then looking, and you see things like this. So in this case, the E. coli that's overexpressing FOH can eat more mannose 6-phosphate, and it actually has more glycerol 3-phosphate. And that's probably really cool because it actually probably goes right here in metabolism. So there's a system when under phosphate starvation where you decarboxylate a 6-hexose sugar, and you turn it into a pentose, and then you use that for getting phosphate. So you get the pentose 5-phosphate sugars, and then those go into the triphosphates, which is exactly what we're seeing in the thing. And we're checking this right now with genetics, and basically what we're finding is that that's how it's working. So we're filling in this thing. So this thing is carrying FOH. FOH is able to, the viruses are carrying it, and it's important for this part of the phosphate metabolism. So now we're starting to do that with a whole bunch of unknowns. And the other thing that we're doing is we've got, we're setting up a crystallization pipeline, so we can just crystallize about 1,000 different proteins that are unknown ORFs to see what pops out there. With that, I'll just thank, there's of course the tons of people that actually do the work. In particular, I need to point out Dana Wilner, who did all the cystic fibrosis stuff, Matt Haynes, and one other person. Let's see on there. Oh, and Nicole Hansen. These two did most of the fecal stuff with Gordon Loud. We've been working with Paul Rainey and Doug Conrad, our main collaborators for doing the cystic fibrosis stuff. And Anka's doing all of those things I showed you with the structural proteins. And then Rob Edwards is my main bioinformatics person that I work with. And then these are the people that paid for it. And with that, I'll quit.
Did you find several cancers associated with HPV, or just one?
If you start looking at the little tumor guys, the unknown tumor ones, and you just use one of those rolling circle amplifications, there's a lot of HPV in those guys. And that's all over. So we just got some tumor from the culture collection and just went out and started checking. And HPV is really common, but they're not ones that you would pick up within the primer sets that people are using. They're extremely derived. There's probably another, there's like, almost 200 HPV now in the database. I'll bet you that's not even the beginning of the HPV thing. There's probably thousands of HPV.
And there is commonly these tumors when they're removed?
Yeah. So all you have to do is take any tumor that you're interested in. I don't know if they're causal though, right? I mean, that's the problem. We've never done anything to really prove they're causal, but take a tumor, do some like mild viral extraction on it, and then hit it with the 529, and you'll probably get an HPV. And I would say that's probably 60-70% of the samples we've looked at. Okay. And we saw that oral, what do they call it, nasal carcinomas. We see those popping up there too. So lung.
And some of the HPV are the same that you find in throat cancer or in a...
So some of them, but actually a lot of them are things that are not known to be carcinogenic, right? So this one that I just showed you in this case has been reported before in a lung carcinoma, even though it's normally associated with a cervical cancer, I think. In this case, it was a cervical cancer. But people have actually found it one time before in a lung carcinoma. So they've got some ability to do that. Yes?
I have a question here. So our microbiome, our bacteria in there, are loaded with these phages there, tons of them there. And yet it is pretty much resilient microbiome as well. So what do you think is the explanation? What's your view on how the bacteria are resisting those viruses?
Right, so I think this is that kind of classical war between lysogens. So if you don't have the virus, you're dead, right? But if you have the virus, then if you get infected, if there's a small burst and that phage comes along, it can't attack you anymore because of superinfection exclusion. Does that make sense? So no, everybody's looking at me like I'm crazy. Okay, so when you have it, when you're a lysogen, right? You cannot be invaded by a phage of a similar class, okay? Which means that you're protected. So what the system seems to be, and this is across the board, looks to be very common, is that you once you've got a virus, it protects you. And then you're living in a community that has a whole bunch of, that have the same virus, okay? And even when one bursts, that virus doesn't kill you. But if you try to invade the system, then you get hit with the viruses that are there and they kill you. This works actually everything up to mice, right? So mice do this. So a mouse will go into, so you have the native mice and then you have a mouse with one of the tumor viruses in it, okay? The invader will go in and it will actually kill all the other mice and take over that niche. Okay? And it's because it's just shedding enough viruses. So it's paying the cost of having the virus, but it's actually not, it allows it to invade a niche. Okay? But once it's there, it can't be...
No defense system or no particular strategy from the bacteria to cope and handle these things?
Well,